Hi, my name is Katrina Derieg. I'm the curator of the Philip L. Wright Zoological Museum here at the University of Montana. We are a research and education museum that can be used by all kinds of people from different research backgrounds. We use the museum in different classes, things like that. And so my role is to take care of these research grade specimens and make sure that they see the broadest use possible. And you can think of my role as kind of like a librarian, but instead of books, it's research specimens.
Hi, I'm Laura Blumensaadt. I'm a third-year PhD student here at the University of Montana in the Ecology and Evolution Department.
And I'm Cheviron. I'm a professor here at the University of Montana Division of Biological Sciences and also the Wildlife Biology program.
So this is my dissertation project. So as I said before, this is my third year, so I've done a couple of field seasons now. And really the overarching point of this project is to better understand the effects of heavy metal contamination from historic mining in the Anaconda area of Montana on small mammals from more of an evolutionary perspective. So a lot of the time when we think about pollution, we think about the ecological effects or maybe more direct effects on species. So, deaths that occur from pollution exposure. But a lot of the time we don't think about it as an evolutionary pressure as well, especially in animals. So, there's been some adaptive research thinking about insects and plants with heavy metals, but not so much in vertebrates. So that's what I'm really trying to cover with my research.
Yeah, and this project kind of builds on a larger theme that we kind of tackle in the lab that's trying to understand how animals deal with challenges in their environment. So we're really interested in what makes certain animals more resilient to challenges. These can be like challenges from harsh environments, but in this case, sort of challenges from human-induced changes to the planet.
Yeah, and we kind of tied the museum into this work because we're collecting specimens that are being archived and can be used for further research. They're collected for this project, but it's really cool that there's more that we can do with them. Yeah.
And I'll also be using museum specimens that other people have collected historically in these areas, both from our museum and from museums across the country. So museum specimens are a super important resource that I'm really happy to also contribute to for researchers in the future.
Kind of like a unique sort of time stamp, right?
Totally.
Of what the environment's like and how it's experienced by animals. Yeah.
Yes.
Yeah, and what we're doing is we're kind of building this infrastructure that someone could go back, in 10 years, 50 years, 100 years, sample in the same place, which is not going to be the same over time, and sort of see how things have changed, which is really cool.
Totally. Like a time capsule of life at different times. Yeah. We've gone out the past 2 summers, we being me and then a team of field techs, which can include anyone from undergrads all the way up to people like Katrina, who are staff at the university. And we've been going out there and trapping small mammals. So we have 2 target species that we're mainly looking at, which are Paramiscus maniculatus, which is deer mice, common name, and Zapus princeps, which is the western jumping mouse. So those are the 2 species of small mammals that we catch the most of at our different sites, and so those are the ones that we're focusing on. And we've been getting a lot of them even at contaminated sites, which is the interesting thing. So, heavy metals have really broad physiological effects, and so it's interesting that they're able to survive in basically normal numbers compared to our clean sites, despite those pressures. So we go out there, we trap mice for a couple of months out of the summer, so mostly June and July, when their numbers are kind of peaking during the year, and we collect a bunch of samples from them. So, and those samples are used primarily for 2 reasons. We use either tissues or a little clip of their ear to get DNA out of for whole genome sequencing. And then we also can use different tissues and blood for understanding their metal exposure, so we can measure metal load in those different parts of their body.
Yeah, and what's cool that we started doing this year is we started collecting parasites as well. So that's my little pet project that I'm interested in is collecting parasites, and then we can compare between clean and dirty sites, you know, parasite prevalence and abundance. So,
Yeah.
we got quite a few ectoparasites. We got some fleas and some ticks, and then a few endoparasites. Not a ton, but some nematodes, maybe a tapeworm or two. Yeah. Yeah. And so and then those are museum specimens, museum vouchers as well. So yeah, it's great.
Yep. Yeah. Very holistic collection.
That's
process,
right. Yeah,
so,
the holistic specimen, the extended specimen in action.
Yes, absolutely.
Yeah.
This could theoretically be relevant for for humans as well. Especially since a lot of heavy metal research has been done in humans because they're concerned, I mean, even in Anaconda specifically, because they're concerned about environmental exposure. And then also, like, another thing I usually write about is just that mining and heavy metals are a really ubiquitous, like, industry and contaminant respectively across the world. I mean, mining is one way that it gets put into the environment, but, like, agriculture, pharmaceuticals, just vehicles are all putting heavy metals into the environment. So, and those are things that can be found everywhere. So it's pretty applicable, I would say, as well. So, yeah. Small mammals are a really critical, central part of the food chain. And so what happens to their populations is going to impact everything up the food chain. The one thing that sometimes people think about that isn't, from what we found so far, not me specifically, but what science has found so far, is that heavy metals don't tend to accumulate in higher predators like some other organic contaminants do. So you might have heard, like, a really common example, which this is a metalloid, but we'll talk about in a second why it accumulates more. In aquatic systems, you've heard about, like, mercury kind of accumulating in, big tuna and whales and dolphins and things like that. And it's actually caused really bad poisoning of people who eat those meats in certain countries. And so that phenomenon is where these metals and pollutants tend to accumulate up the food chain, because the higher order predators are just consuming so much of it that it's really concentrated in their tissues. And that's, with mercury, it tends to be because mercury gets chemically changed to be methylmercury very often, and then it acts more like an organic contaminant, which tends to bioaccumulate more. But or biomagnify more, I should say. But with heavy metals, we don't tend to see that. However, it's still transferring through the food chain, so it might not be accumulating in amounts that are actually leading to deaths in those predators, but it can still impact them through toxicity. So, we've heard a lot about lead toxicity. Cadmium is also a neurotoxin, and those could all affect things like their ability to predate and their ability to find other individuals to mate and things like that. So could certainly still affect their ability to survive. But yeah, and then, as I said in the beginning, also just if mice are dying a lot, then there's not going to be as much food for the predators. But if they are able to adapt and they're able to just survive on that landscape, then there's still going to be abundant food for other members of the food chain. So there's a few different kind of indirect ways that it can affect it. But
cascading
effects, yeah.
How are the mice getting the heavy metals? Is this through the soil? Is it through the water? Is it the plants that they're eating? Like, how is it getting into their bodies?
Great question, and all of the above and more. So, specifically with mammals, there's 4 main ways that they accumulate metals. So those 4 ways are, they can get it through the placenta and through lactation, so from their parents. So before they're even born, they can be exposed to it. Once they are born and they're out interacting with their environment, they can inhale it from the air, which was certainly more of a problem when the smelter was actually active, but can still get kicked up in dust and things like that. And speaking of that, with mice in particular, because our species that we're looking at are fossorial or semi-fossorial, and they're digging in the dirt a lot and they're burrowing underground, they can, both accidentally ingest soil particles, both just from that digging and from the food that they eat, and also just kicking up dust from the digging process. So, and then the other thing is that they're also constantly in contact with the soil, and the soil is super highly contaminated, and metals can be absorbed through your skin. So just that constant, physical contact with contaminated soils as well can really be a problem. So, yeah.
And when I think about the significance of this project, I think about how where we were trapping in Mount Hagen Wildlife Management Area, it's also a really big recreation area.
Right.
And so, I mean, so it's a place that people want to be and maybe don't know that,
Yes.
that there's, you know, and not that that the heavy metals are posing, you know, these immediate direct risks to people out there, but I think it kind of shines a light on this region that also is impacted by so many other factors. So like disturbance, you know, I mean, that's also going to impact populations of small mammals and where they can go and what they're going to eat. And so I think there's just so many like different ties in this area. That's what makes it a really good study site, right?
Totally.
Right,
Yeah.
right.
Yeah, definitely. I mean, Anaconda itself Anaconda and Butte have been, I've seen several studies that are specifically focusing on people in those areas to see whether there are higher rates of different diseases and things like that, and there are. And these sites are just a few kilometers away from Anaconda and certainly highly impacted without any. There have been some restoration efforts in terms of replanting plants there that are more hardy and resistant to heavy metals, but they haven't actually gone in and removed the soil and removed that contamination. So certainly I would say it's a concern for humans that are recreating in that area, and people should be cautious about that. But I mean, go a few more miles up the road, it's not too bad. So,
But along these lines, if we can understand sort of the physiological processes that allow the mice to kind of survive and thrive in these environments, we might learn something about human health, right?
Totally.
Like the things that could impact, you know, human. The ways in which they can sort of deal with metals and things like that. So just kind of understanding the basic physiology that allows them to deal with these metal loads is interesting from a biomedical perspective.
Definitely, yeah.
Yeah.
Because we can delve in certain ways a little bit deeper with using mice than we can with humans. There's always going to be limitations to human studies, so it's great to be able to use other mammals that could have, you know, very relevant information to give us. So, yeah. Very first thing, just the numbers that we got out there compared between our different sites. I mean, we were expecting, you know, even if, let's say, the mice are really highly adapted to high levels of metal contaminants, we would expect the population sizes to be relatively equal between our contaminated sites and our uncontaminated sites. But these mice are magnetic, and they love heavy metals, apparently. Not literally. But yeah, we've just been getting so many more. It's so much easier to catch mice in our contaminated.
They seem to really be thriving in these places that you really wouldn't expect it, right?
Yeah, which is really interesting. Yeah.
And some of that might be related to the fact that they, on the most contaminated sites, they're sort of the only thing there. So they're kind of free from competition from others. So maybe they're like evolutionary adaptation to exist in these habitats frees them from competition, and that allows them to kind of thrive in these places. But really surprising how common they are relative to like control kind of clean sites, right?
Yeah, I mean, we struggled to get mice from clean sites.
Sure.
You know? They just loved being in the dirty sites. You know, we kept going back to clean sites and setting a bunch of traps and, you know, get 3 mice, you know?
I mean, that's kind of a deer mouse thing, right? Like they do love and kind of thrive in these marginal kinds of habitats, you know, not just metal contaminated sites, like mountaintops, you know, they occur on the floor of Death Valley, right? They really kind of enjoy disturbed and harsh places,
Yeah, that was what kind of surprised me, was how few deer mice we were getting, but how many jumping mice we were getting. Because jumping mice, they're kind of habitat specialists. They're in this tall grass, mesic, you know, kind of environment. And deer mice, they can be sort of wherever. And so I just kind of got me thinking, I wonder if because there's so much great habitat for the jumping mice that they're just so dominant, and the deer mice, they're not going to compete for the great habitat when they are able to live in the crappy habitat. And so why would they try to compete with this, you know, bigger mouse that, you know, is specialized? So it's just interesting, just kind of from like a natural history perspective, to be out on the landscape and not know what to expect.
Because I mean, we think about deer mice, they're this great, I mean, their genus is this amazing system for studying adaptation and has been used for that in a lot of different contexts. But jumping mice have barely been studied, and yet we're seeing them in these equivalent numbers on these contaminated sites. So I'm like, what does that mean about their ability to cope with these environmental stressors and maybe adapt to them? So I think I'm really excited to look more into that further.
I think it's also worth noting what's cool about comparing these two species is that they are not closely related rodents.
Not
They are
at
in
all.
totally different families in different parts of the rodent family tree. And so I think that makes them super interesting to compare.
Well, and if we find, mechanisms that allow them to tolerate these metals that are the same across those deep evolutionary timescales, then that can tell something about like a general mechanism, a general way in which mammals can deal with, high metal loads. I mean, another thing that was surprising is just how much metal these mice have in their bodies, right? It's, it's toxic
several
levels.
orders above what, we would consider toxic in humans. It's pretty
pretty
impressive.
intense.
So I wonder if any kind of illness or disease can be observed from the metal contamination. I mean, we didn't see any mice that looked like they had issues with them, right?
Yes, right.
But that's,
Yeah, it's hard because it could be subtle things that aren't going to be immediately apparent to us within the 5 minutes that we have a mouse in hand. So, but in the future, we kind of want to look more into that and do some more specific tests for different physiological effects. So it'll be really interesting to see what pops up there.
Yeah, so what do you think about with these experiments? Right?
This goes into kind of our future plans, but in the future, we're thinking about having mice in captivity from these different sites and doing some experiments looking at physiological response to metal exposure and just comparing to see whether mice that have been living on these sites and for generations seem to be having a reduced response or reduced negative response compared to naive mice, so to speak. And just that would be somewhat of an indication that they've started to adapt to these metals and just aren't experiencing the same sort of physiological effects anymore.
And one of the major effects of metal exposure, of course, is effects on cognition, right? And so you have some ideas about kind of testing whether or not the metals affect their cognitive abilities.
I mean, I'm sure a lot of people have heard about like lead toxicity from historic stories. That's probably one of the more popular known ones, but a lot of these metals have neurotoxic effects, and so we're thinking about doing some cognition experiments where we're looking at things like memory and how well they can remember different spaces that they've been in before. Things like anxiety we can test as well and see how willing they are to be in open spaces versus enclosed spaces. And so there's different things that we can do to look at cognition.
I'm so excited to see the results that you're going to find in that. I just think it's going to be so cool.
Yeah. Great question. That's a question we all want to know the answer to. I mean, I will probably have at least another 3 years, including this year. Whether it ends up going longer will just depend on how experiments go, and a lot of science is luck and things falling into place and seeing whether there are any barriers, just all kinds of things. So, but probably about 3-4 more years. Yeah.
For sure. I mean, the thing about science generally is that projects generally don't end, right? Like, you learn something, you continue down a line of inquiry, and so I suspect that, even when Laura leaves here, right, she'll continue to follow this kind of line of inquiry wherever she ends up being a scientist. We'll probably continue doing similar things in the lab. It'll branch in different ways that we can't even really predict. So I wouldn't be surprised if we're working on this problem for the next 15 years or so, in a combination between Laura doing her kind of own independent thing, us doing kind of parallel complementary stuff.
Totally.
Yeah, and the museum is certainly here to stay.
Yes,
so,
absolutely.
And so that really kind of roots it in this longer term thing because we've got these samples and they're archived, and so they can be re-examined with new methods in the future compared to new samples collected. And so that's what's really cool. And this is the kind of project that really lends itself to being a long-term project.
Absolutely.
That's the beauty of it, right?
Totally.
So we want to be able to see that change over time.
So if someone wants to continue this research in 100 years,
Exactly.
they absolutely can, because these things will all be preserved. So
that's the power of museums.
Power of museums, Totally. so I will be using historic museum specimens in my research to look at genetic changes over time, over the course of the timeline of mining and the smelter being up and active. And so there are a handful of specimens collected in this area from all the way back in the mid-1800s, before the smelter or mining was even happening, which will be really interesting to have specimens to compare before that selective pressure was ever around. So,
Totally irreplaceable. I mean, it's
Yes.
just like this sample in time that we can't get again. And, you know, so, yeah, it really highlights the importance of natural history museums, right?
Yeah.
You know?
Yeah, especially because these specimens were collected long before we knew that this would be an
Right.
issue.
Yeah,
Long
exactly.
before the technology to test for this existed.
Yep.
And so that just kind of highlights the same kind of thing that we're doing right now, is collecting things, and this is a baseline for this place at this point in time.
Exactly.
Totally.
And yeah, so you can't go back in time and sample. This is the closest thing we get to a time machine, right?
Totally. Yep.
Some clever grad student 100 years from now is going to have an idea that hasn't even occurred to us. Right?
Exactly.
For
Yeah,
their
with
education,
technology that just we can't even dream of.
Yeah.
Yeah,
Yes,
which
absolutely.
is really cool. Yeah.
Yeah. We have to answer this
Yeah, and I
all
think
the
it is
time.
Yeah, I kind of
Yeah.
want to like preface this that, you know, in science we always have to defend what we're doing, why we're doing it,
Why we
why
should
it
have
matters.
money.
Yeah, why a funding agency should give us money.
Yeah.
Yeah, it's super critical.
Yeah.
Yeah, and I mean, that's reasonable, right? Like,
Sure,
this
yeah,
is
of course.
Totally.
taxpayer money, right?
Yeah.
So we should be responsible with the way we're spending that money, and we should be able to justify it and like kind of answer the question.
Agreed.
So yeah, what do you think? Like, what would your answer to that question?
Totally. I mean, I have a few answers to this question. I mean, just for, you know, I think like understanding the limits of adaptive capacity is super important in our world today. Like, things are changing so rapidly in so many different ways. And what does that mean for the trajectory of wildlife across the globe? Like, that is just a fundamental question that so many people are
don't know
trying
the
to
answer
answer.
to.
And this is just one way of looking at that question, right? Thinking
Yeah.
about it from this evolutionary perspective, which can give us insight into both the past and the future.
Yeah.
So I think from that standpoint, yes. I think also just, you know, as we've said before, you know, thinking about like the connection to human health and the relevance to that. Again, humans are exposed to metals a lot from, you know, not just mining, but all kinds of industry.
Yeah,
I mean, if
yeah.
people are living near industry, even just near major roadways, that's another source of pollution, just things like that. It's important to understand how that's affecting us on in different ways. And yeah, I think just it's another, I think this is attacking this question of pollution and ecological disturbance from an angle that is less commonly studied. We think about it in an ecological way, and we, you know, ask these questions about, you know, what the death toll is like from pollution events, and you know, you see the oil spill videos of them cleaning off the little baby ducks. But this is kind of taking it in like a more nuanced direction, and thinking about the effects that aren't as apparent to just the naked eye when you're looking at these things, but that are really going to affect populations a long time down the line. So
Yeah, I mean, it's like it's an open question as to how much or how quickly wildlife populations can adapt to like changes we're making to the globe. And so Laura's work is getting at that question directly. Like, can on these really like decades-long timescales, can animals adapt? So, and then how, right? And once we understand how, like, yeah, there's biomedical relevance to that. So, I actually think that this is kind of a hopeful story. Like,
That's what I was going to say. Yeah.
we often start from the perspective like, well, you know, pollution, lots of wildlife populations are in decline, you know, and it looks bleak. But here, this seems to be a case where we have really rapid adaptation to a really dramatic stressor that we've imposed. And it just suggests that, you know, populations, wildlife, pretty resilient, right? And this kind of tells us a little bit about how that resilience works and how fast it can kind of change. So, yeah, I find it pretty hopeful, honestly.
Yeah, and
I
I think
agree.
that's the direction that a lot of research has been trying to go,
Yeah.
because there's been a lot of doom and gloom
For sure.
in science for
sure.
many decades,
Yeah.
right? And sort of these looming threats, but to be able to say that nature's pretty resilient
Yeah.
and has dealt with a lot of
Lots
things.
of
Yeah.
challenges.
Yeah, a lot
Nature
of
finds a
challenges.
way.
Yeah, that's right. Nature finds a way, and
Yeah.
so yeah, I think that's compelling to people, to the public as well, that, you know, we're trying to spread a message of hope, and that you know, and through doing this research, we can actually be able to say that and not
quantify it and understand how it happened. Yeah, exactly.
Totally.
Yeah, I agree.
Yeah. Yeah, so this summer, Katrina wasn't there this day, unfortunately.
No, I wish.
But I was out there with two of our other amazing field techs, and there was one site that we had where we were trapping around these kind of old buildings, and there was this opening underneath one of the buildings, so you could, like, really see underneath it, and one of my techs was walking around checking traps, and she was like, there was one that was right here, and it's missing now. And so after a little bit of looking around, she realized that it had been dragged about 10 feet under the building, completely out of our reach. And so, and our culprit, who she saw probably 10 minutes later, was this very adorable yet very mischievous weasel who was standing.
It's always the weasels.
It's always the weasels. And so we then had to use some creativity. There's a lot of creativity in science. You'd be surprised. A lot of problem solving.
Especially field work.
Especially with field work. And we scavenged some PCP pipe and barbed wire from around the house and managed to hook, like basically chopstick/hook this trap out from under the building, and we did get it back, and it was fine. And there was nothing inside of it, so no reward at the end either. But we did rescue it, so that was one of our fun stories.
And I was particularly grateful because these traps are more expensive than you would think.
So, more expensive than they should be.
More expensive than they should be. So I was appreciative that all the museum gear made it back.
Sure.
Yes, absolutely.
Sure.
Yes, yeah. And then our first year, we had one site where there was a harrier pair that was always, you know, hanging out around and flying around our site. And one of my poor field techs was going and checking traps, and the vegetation in the site is super dense. And he stepped, and all of a sudden a harrier just came flying out at him, screaming from the bushes. So he definitely had gotten too close to her nest, unfortunately. And she would not leave him alone for the rest of our field season, actually. Yes. So that day, I just kind of heard like a yelp from far away, and I was like, what's going on the walkie? And he was like, yeah, I pissed off a harrier. And so then I met up with him, and we hid in the bushes for 10 minutes, just literally laying on the ground so that she couldn't see us because she was so mad and kept trying to dive bomb us. And then I think we went back to that site a couple more times during the season, and she would just start screaming immediately upon us showing up.
She remembered you.
She remembered, yeah. She was not happy, and we were like, we're sorry, we're not here for your babies. And, you know, we're also taking her food, so sorry, we're coming in and disturbing you.
Not too much.
Not too much. We happened to be walking in the same area, and that was enough for her to be pretty upset. So, yeah.
That's
Dad
funny.
didn't care all that much. He was just kind of hanging out, but yeah.
I hadn't heard that story. That's funny.
Yeah.
Yeah, it was a good field season overall.
Yeah.
It was a lot of fun. There's a lot of camaraderie in the field.
Yeah,
you know? I mean,
totally.
It's so fun.
Yeah. It is a job unlike any other, I will say.
Yeah.
Mm-hmm. But super fun, and it takes a lot of preparation, but it's the part that we all like the most. So, yeah.
That's true.
Yeah.
Yeah, that's why people, I think, sort of in our fields, why people become biologists, right? Is to be outside, you know, with the animals.
Yeah. I think your first time doing field work is always pretty memorable, and you're just like, wow, you actually get to be outside and do this as a job. Like, that's amazing. That's so cool.
I think it's also really satisfying when, you know, you get your data and you know that those data are from a mouse that you caught, that you went out there, you trapped that animal, and now you've got the data from it, and you analyze it, and you publish it, and it's just, that whole process, start to finish, is incredibly satisfying.
Yes.
And the moment that, like, you know something that literally nobody else in the whole world does. Like, this is a new discovery that you've made.
Totally.
It's just a really exciting, you know. It's just, you can't replicate it, right?
No, you can't. Yeah, even before doing my own project, I feel like working on other people's projects, I was like, no, I want to be involved in every part of this, because it's just so satisfying to see it through beginning to end and know okay, this is where this number came from. This is where this mouse came from, you know, and you were just there for all of it to see the whole thing. And it makes it so real. It's not just numbers on the screen.